Good morning, everybody. Happy Friday and welcome to the Friday UAB Informatics Institute Power Talk series. I'm very happy to introduce today's lecture. Dr. Jake Chen is very well known to us, Chief Bioinformatics Officer and Associate Professor at the UAB Informatics Institute. He's also Associate Director, Professor of Genetics with appointments in Computer Science and Biomedical Engineering. His areas of focus are bioinformatics, systems biology, network biology, artificial intelligence. He earned his PhD in computer and information science at University of Minnesota Twin Cities, and was also on faculty at Indiana University in bioinformatics. He's founding director of the Indiana Center for Systems Biology and Personalized Medicine, and also he's played a great role in UAB in data commons and data sharing and access to data resources and analysis. He has over 25 years of research experience in biological data mining and systems biology, over 180 peer-reviewed publications, and he's an elected fellow of the American College of Medical Informatics, ACME, and the American Institute of Medical and Biological Engineering. And he's also on the editorial board of BMC Bioinformatics, Journal of Medical Informatics Association, and Frontiers in Artificial Intelligence and Big Data. In 2019, he was recognized by Deep Knowledge Analytics as one of the top 100 AI leaders in drug discovery and healthcare. Zhandos Sembay is a software developer within the UAB Informatics Institute. He earned his MS, Master of Science in Computer Science from UAB. And he has a great role in helping with the developing applications and to allow people to access data for research. The title of the talk, Acing the New NIH Data Sharing Requirement with U-BRITE.
Well, thank you, Amy. I think if you're here today for a research talk, you'll be disappointed because today is all about you as an NIH investigator looking for some kind of a help from the Informatics Institute. So I'm primarily wearing my hat as the Associate Director of the UAB Informatics Institute, joined with two of my colleagues, Zhandos Sembay and Dr. Zongliang Yue. Especially, I want to honor my appreciation of all the work for Dr. Yue, because he will be upgrading his career to Auburn University to join as a research assistant professor very soon. With all of that, let's get started. So it will be a three-person presentation. As we mentioned, that the three of us are here as your quote-unquote instructors, and they will be very practical. We'll teach you some basic concept, and this is a division of work. Initially, I'll talk about the data management and sharing plan. What are the 6 different elements that you need to pay attention? I'll give some examples to really get you to understand behind the scenes what NIH is really looking for. And then Zhandos will tell you, hey, if you want to spend money elsewhere, getting company to support it, yeah, it's fine, it's your choice. But UAB currently, we have a free option to develop your DMSP, and it's called U-BRITE. And we want to tell you all the components of U-BRITE, and he will just take care of this part too. And Dr. Yue is currently working with me on various bioinformatics research project. He will actually walk you through one real-world R21 research proposal, and then he will carve out sections of the data management and sharing plan from the proposal submission to make sure that you understand, even as a top-of-the-line bioinformatician, we have a great template. And this may not be addressed by the minimal free option of the U-BRITE, because there's a lot of thoughts and coordination being put in. But at least it gives you a spectrum on what is the minimal and what is that we can do as an advanced option to ace this writing. So roughly, I'm going to spend 15 minutes. We're already 5 minutes into the talk, and talk about the 6 elements. Zhandos will spend about 20 minutes. And then I'll switch over to Dr. Yue, talk about the case study for 15 minutes. We'll have questions and answers. And Zhandos is going to share in the chat already pre-filled out the question and answers that we collected from previous sessions, thanks to UAB's Office of Research Administration. They have already had some wonderful sessions that's well attended by more than 100 investigators. And so we took that sheet and we thought, okay, why don't we answer it from the U-BRITE perspective? So if you click on the link, you can comment there. You can also put your comments in the chat box in the last 10 minutes, and we can answer additional questions. All right. So let's get started. I like to make it interactive. If you can use your phone to scan this QR code, or just go to your browser called slido.com and then enter this 7-digit code, you'll be able to answer 2 questions. One of them is here. What made you interested in today's webinar? Now I'm gonna give everyone 60 seconds. Just go to slido.com and enter this and see if we can have some answer. I see one person already answered. This is a multiple choice, and so far most people I can see are interested in seeing what UAB professionals are doing to address the data management challenge. People do have some concerns that, about the loss of ability. There's no correct answer, and this is just to get everyone engaged. We more or less see the trend, 28, 30 participants into this. Wow, this is almost on par with my expectations. I wrote it in the order of my perceived preference, what is more important in people's mind and what is least important. But nonetheless, the concern is also there that what if we share the data and we lose the control as a biologist? Is our field moving into something that is darker than is necessary? Okay, so I'm gonna move on. This is an NIH scientific data sharing website. There are lots of information about how to write data management and sharing plan. Of course, it's required if you submit your grant after January the 25th, even if it's a revision of the previous submitted proposal that doesn't require the document. And I'm not sure whether they will make it a required scoring criteria, because as many of you know, that in the NIH grant review panels, there are data sharing, resource sharing usually is not the score driving discussion, but people can raise concerns, and then it's up to the program officer to determine whether to put yours on hold when there are 2 excellent proposals with the same score. So it's very important to read every information about this. Now let's do another poll. Go back to the slido.com. And this one is open-end questions. So why does NIH require a data management and sharing plan for every grant proposal now. So I would like you to open up some comments. Do you think that they're doing it to benefit only informaticians, or they have some kind of a controls? And what do you think? Yeah, one more hoop to jump through. Yes, I like that. It's anonymous. Feel free to open up, and Congress mandated. Transparency of data, that's promote healthcare reproducibility, public funded research. I see great comments, reuse of data, reproducibility. Yeah, where many of you are right on point. We are living in a world where every dollar matters. If only one group benefit from the data that's generated is one thing, but if 100 groups outside of your own can also look at the data and combine it with other data set, it seems to be a wonderful use of the federal dollars. I think there are excellent comments, and all of this will be just captured. It seems that the most people mentioned things about data reproducibility is and encouraging research. That's been a general trend in our replies. Okay, I think this is great. This is my answer. So I think I guess it's why NIH requirement for separate plan is intended to promote good scientific practice, encourage sharing of research data, ensure long-term preservation of research data generated for studies. So there, I think essentially it's a three aspect. One is a long-term value of the data will go beyond the life cycle of one's own grant. And so they want to start thinking about that. And then transparency is really good to maintain rigor and reproducibility, even if there's like an irreproducible result down the stream, because all the data are shared, and then we can track back to see, oh, maybe there's a special condition. They were using a different mouse model, and therefore the result does not translate in humans. And also in biomedical informatics, we emphasize a lot about the FAIR principles to make sure every tool and the data resources that we develop can be findable, accessible, interoperable, and reusable by the community of researchers. So all of this make NIH want to just require this plan. Now it comes to the 6 elements. I'm going to actually elaborate on some examples on the 6 element, and the Zhandos will repeat it from the U-BRITE perspective, and Zhongliang will also use the 6 element to talk about his case study. So it's very important to understand what are these 6 elements. So the first one is about data. Let's see. One very important concept here is NIH wants you to become expert custodian of data and also your metadata. Data is relatively easier to understand. The data can be genomic data, metabolomics data, transcriptomic data, imaging data, social economic survey collected data, environmental data, electronic medical records, behavioral information, medication, anything that that you collect from your study can be considered data. Even though we say this is big data, but it doesn't have to be the big data. It can be documentation, publication as well. Then one important distinction is there should be metadata captured. Metadata sounds very mysterious, but when you think about it, we use metadata all the time. For example, if you have a data set, and if you want to describe about this data set, you say, what is the title of this data set? Maybe you're talking about cardiomyocyte single cell analysis. That's the title. Who are the authors and what are the genre, maybe single cell transcriptomics, publication date. And this in itself is one type of metadata. There could be technical metadata that talk about size of your file, whether you're compressing it or not. There could be metadata used for preservation. Checksum is used to check for integrity of the file. If the file has been edited or tampered with, the checksum will be different. There could be rights. Do you want it to be openly shared or with some kind of a licensing agreement? Who can access your data? That's the rights metadata. There could be structural information, right? So if it's an ontology, what position of the ontology is it located in? If you're using markup languages, and you can also specify the structure of information. So all of this information you need to now think about. The second element is about the software tools. And software tools today is very different from the software development even 10, 20 years ago when I was just a junior faculty or just a PhD scholar. Most people are using open source software development, so you need to think about using GitHub, or the UAB equivalent is a GitLab. So there are 94 million developers, and then 90% of the companies use open source development. More than 90% of Fortune 500 companies use GitHub. And there are 413 million GitHub contributions that are project. So it's very significant community that I would say produce more than the entire biomedical abstract or just publications in the world. This is one example of the GitHub instance of a software that we just published in this journal. Last night, I saw that the citation wasn't there. I just edited the file to include the citation. So you can certainly put documentation, but more importantly, you can put your source code all openly accessible to the public. UAB's version of GitHub is called a GitLab. It allows you to keep the code private until your publication. So you have more control there. And the third element is called the biomedical standards. We're a genome data scientist, and this is one example of data standards called the gene ontology. So it's more of a directed acyclic graph where the nodes represent terms that scientists can use to annotate gene functions. This is the human phenotype ontology, and that's research scientists' way of looking at diseases and the classification of diseases. So it goes from general to specific term. It's an atrial septal defect, and then there's a hierarchy of terms. HL7 FHIR is a data exchange format for electronic medical records, and it's gaining a lot of fire because this standard are adopted by health IT companies throughout the world. So you want to talk about the standards if your data release conform to the standard, that really would make it easier for you to plug into the community of researchers and vendors. And then the fourth element is about the data preservation. Essentially, the data preservation, if you don't have a capability of developing databases, you should select a public data repository and make sure that your data set have the identifier you can document in your manuscript. Or when the NIH officer asks you, you can just build a table of identifier and submit it to your annual report. So examples of the data repositories are protein data bank, if you have the protein structures. If you have functional genomic data set, you submit it to Gene Expression Omnibus. If you have the clinical cohort and GWAS study, you can submit it to dbGaP that have very good data use agreement in place to guard the controlled access to your data set. National COVID Cohort Collaborative is a national effort to contribute all kinds of data in an online data science platform that is much easier to use than dbGaP. U-BRITE is what the Zhandos is going to talk to you about. And the pager is our own software. So if you are like informaticians, you don't want to submit it to the public databases and your data is so new, then you can build your databases and manage it yourself. The fifth element is about the controlling data access. Not all the time that you need to, you want to actually have entire data set freely available to others because maybe informed consent or because you have a patent pending, you don't want to release the chemical compound structures until a later time. So for controlled access, you can actually define data use restrictions and requirements. And there are actually even ontology that describe what you need to consider in order to build up your data use requirement document. That's you as a data depositor. And then you can also define who should be eligible to request your data. Maybe not high school teenagers, maybe not the criminals, but probably your competitor will want to be requesting your data. You can't, there are certain rules that define what you can or cannot prevent people from requesting your data. And then usually the data repository should have some kind of a matching algorithm. This is a scheme that is adopted by dbGaP database, by the way. So they will be able to manage the complex scenarios of matching data requesters and data depositors. And the last element is the data oversight protocols. There are many things you need to think about. How do you manage your data to ensure security, backup recovery, archiving, preservation, data sharing policies. It turned out that this will come as a cost. If you don't have too much data, then maybe you can do it for free on any system. But if you found that you have very complex data, lots of modalities, tons of them, and you're generating them on an ongoing basis in a consortium, you may want to find the qualified informaticians who can do it proactively to ensure all of that. Okay, and I'm gonna stop sharing and let the Zhandos to start his part.
Yeah, thank you, Dr. Chen. So, hello everyone. So now we're gonna use to the next part of our agenda today. So the first, I would like to start from introducing what's U-BRITE. So U-BRITE is our biomedical data science infrastructure built by our informatics institute teams and supported by research computing group also. So you can visit our ubrite.org website to get more information and find the NIH data sharing plan related section. And also, so currently we have managed over 300,000 managed files. We have more than 40 team projects, and currently we have about 200 users. The main goals of U-BRITE is bridge gaps between translation research and data science advance, also advanced informatics tool development towards clinical multi-omic studies. We're trying to help build some stable interdisciplinary research team. So also U-BRITE provide biomedical informatics, data science, and AI training. As an example, every year we have the data science hackathon, which is supported by U-BRITE team. And also we support U-BRITE hosted data, linking, exploration, navigation, and search for fair compliance. That's our tool, U-BRITE Data Lens. So I'm gonna talk about Data Lens later. And so how actually U-BRITE can help for data management and sharing plan requirements. So here you can see that I divided our U-BRITE architecture tools in different 6 elements. So U-BRITE currently use different tools such as the GitLab, GitHub, the Python, the Globus. We also use UIB Chiha supercomputer, the box folder. We also have the Zhandos community. And as an example, we have I2B2 tools. And yeah, let's walk through in each element. First, we start for element 1 is about data sharing and about data hosting. So currently, U-BRITE data repository is implemented on top of managed high-performance parallel storage connected to our UIB Chiha supercomputer. So where researchers can interact with their data, which is often made available as a flat files that fit this abstraction, or discover other locally cached omics data of interest here in this repository. So we have our U-BRITE directory on Chiha. So we also, you can discover some omics data, and the list of omics data you can find on our website. And for documentation repository, we use the box. The box is implemented with UIB Research Computing Group. So the box provides an enterprise cloud platform for secure content management. So in particular, that's a shared files and folders abstraction that should be familiar to most investigators. Desktop integration with Microsoft Office, Boxing, and Box Drive, and the flexible sharing and permission models that facilitate team collaboration. And the next element is about tools. So as an example, GitLab and GitHub. So currently U-BRITE, the source code repository is implemented on UIB local instance of GitLab, and that's maintained and supported by Research Computing Group. So here you can find the links to our GitLab group and GitHub. So right now I'm going to show just quick demo on our U-BRITE GitHub community. So yeah, you can follow us on GitHub. Currently we have more than 60 members, and you can find some projects from last year hackathons. And as you can see, so most of our users use the Python programming languages, R, and also we use Jupyter Notebook, HTML. And yes, you can follow us. And also we have the U-BRITE GitLab community. So that's this group is for teaching and learning oriented namespace for U-BRITE. So where we store the demo and some tutorial project, and as well as for hosting team contributed project from different info classes, NIH data management plans, and the hackathon events. So you can request the access for our U-BRITE GitLab community. Yes. And the next tools is about the access how to access the data on Cha. The first one is the Globus. So the Globus is a powerful tool for robustly and secure management, data transfer, and also sharing. So data transfer to and from the collaborators and within UIB research computing. The second is FileZilla, is actually as a free SFTP platform. Use it to transfer data between our local machine and a remote server. In this case, so remote storage accessible to our UIB Cha supercomputer. And the next element that's about the standards. So as an example, we have the UIB Foundation Ontology that's built by our informatics team, Informatic Institute team. So that's actually the human phenotype ontology that we supported for AHAI, and gene ontology that we support for genomics. If in your data management sharing plans there is no standards, so you have to know this information and build your own standard with the data dictionary. And so, and next is element about this identifying and the data. So, and data. So to ensure the long-term accessibility of your data or your source code, we should copy of the GitLab code repository or GitHub code repository and your public data. So will be archived in U-BRITE Zenodo community at the time of publication. So what is Zenodo? Zenodo is an open access repository that's specialized in preserving software and issues DOIs for code. So the code DOI will be included in each resulting publication. So right now I'm going to show the demo of our U-BRITE Zenodo community. So yeah, if you go to zenodo.org communities/U-BRITE, so we have the good community. So we use this Zenodo community as a namespace for keeping tracking of our various research outputs, including, but not limited to, publications, poster, presentation, datasets, software, and some training materials that produced by our research team and also supported by U-BRITE team. As an example, we have the pager. So the pager, so currently you can see that the authors and some acknowledged about, and then and we mentioned about U-BRITE location. Actually, this data stored on our U-BRITE location on Cha supercomputer. So, and for keeping tracking and identification, so you will get DOI to identify your data or source code. So, and so we have different data sets that we local cached in U-BRITE. So that's some about the mammogram breast cancer as an example. So here you can see the location. Also, Zenodo is good for keeping tracking of how many views and downloads. So you can see some short statistics, how many views and how many downloads. And you can easily get the credits to your work, and somebody can cite your work. Yeah, that's about the Zenodo community. So you just need request the access to upload it in our U-BRITE community. Then our team will accept it and we will host it on our U-BRITE namespace. Okay, so that was an example of using Zenodo for keeping in U-BRITE. So, and also for right now, we developed U-BRITE Data Lens. That's a tool for that's a good tool for data management. So currently it's still under development, but we hope we will release to public very soon. So yeah, here's some about demo about data lens tool. So you can see there's some statistic about this overview of research data available. So about some volumes and how many statistics about the data. So, and to keep tracking all your data, you can create your project on U-BRITE data lens, and you can see keep your protocols, your documentation, some clinical data sets, code repository, so your grants publication in one place in U-BRITE data lens. That's a good tool for data management. Yes. Again, that was just quick demo of U-BRITE example. The next element is it's about the privacy. And so as an example, I would like to show you I2B2. So I2B2 is a tool implemented within UAB to enable effective understanding of UAB patient population and to facilitate access to data and support of research. So here's an example of the access of I2B2. So the access to I2B2 is available to IB personnel with Blazor ID, but also requires some validations. And upon completion of the access request, will be verified the following. You have to hyper training, RB, human subject protection training, and some acknowledgement of restriction on use of limited data sets, and supervisor approval from non-faculty. That's a good example of element 5 for accessing data and privacy. So, and let's move to another, the next element is 6, is about the oversight of data management and sharing. So if you're planning work on your project using U-BRITE, so all researchers can access U-BRITE resources and tools through 2 tracks. The first track is basic, which means the self-serving, and then advanced, that's supported by our team. So if you are in need of informatics support to use our U-BRITE and tools, so members of the Informatic Institute and also in collaboration with the Research Computing Group are available to assist for fee through the support track. So if you don't need informatics assistance, you may access U-BRITE through the self-directed track. Here's our team, our informatics team, and also research computing team. So if you would like to get started project with U-BRITE, just visit our ubrite.org get started. So, and you can find more information how to get help from our team. So you can contact Dr. Chen, and you can also contact with me for technical support. Okay, so next is about the final. So what do you need to work in U-BRITE? Here some U-BRITE readiness checklist. The first is about accounts. You should have some Blazor ID. Also, Chiha account has access to U-BRITE GitLab account. And for data management, so all big data we store and manage on Chiha. So here's location on Chiha. So, and that's the permission is restricted only for U-BRITE Unix group. So you can ask the access from us. And also all project files should be committed on U-BRITE GitLab and GitHub group. So, and all your documentation upload to U-BRITE box, and all code repository and public data achieved in U-BRITE Zenodo community. So that was quick overview of each section using U-BRITE. So, and next is in our agenda, Dr. Yu.
Okay, so first I want to introduce about the in consider of those slider collected most concerned terms like the data management as well as the reproducibility. I'm going to just have a real world bioinformatics R21 proposal, the MSP example, and here's the background. We previously have a pack that defined as a list of the genes plus the curation. We also have this option information such as the weight, the structure, and data links. For the pack, it's an abbreviation for the pathways, annotated gene list, and gene signature. So pathways always have those very detailed information about the signal cascade, and annotated gene list is from the computational generated results such as RNA-seq, microarrays, proteomics. And for the gene signature, this is like a raw data have been generated from those sequencing data, but without any annotations from biologists. So therefore, we have another idea about how we can write some grant regarding to super pack construction. And we also mentioned about the related tools and data. So in the packs, they are not isolated. They actually have been connected to each other through those co-membership and regulatory relationships. So therefore, we have this pack-to-pack network constructed. And next, we want to just extract those dense super packs. You can think about like a super cluster of the packs by defining this packs cohesion and also this density structure that we extracted from the previous network. In the stage of this data generation, we can have 5 major types of the major output of these tools. So the first is about the compiled and executable model code and configuration files. The second is for the result related to the performance and the quality from the scientific aspect. And for the third is analysis result from prototype runs. And fourth is a side of the real world modules for PIs. And the fifth is the papers, posters, and talks related to the dissemination of this work. So we also form, according to the NIH guideline, to form a 2-page data amendment and sharing plan. Just divided into this 6 elements. The first element is about the data type. Particularly in this grant, we have all those data, like in-house data, and also public available data from U-BRITE stored in the UAB Chiha supercomputer plus server. And we also have those pager web server and the real world data site downloaded or generated by ourselves. So where we have this synthetic superpack data and real world superpacks generated by the developer and provide this structured network structure. All those generated, all those superpack modeling code will be deposited in GitHub repository, and in stage code we're going to store in the GitLab UAB. And for these 2-dimension or 3-dimension of the single cell t-SNE plot or UMAP plot, and also those row format, generated from single analysis, these in-stage results are deployed in the UAB server to be loaded to an interactive web server that will be convenient for our investigator to get a view and also refine the result accordingly. And for the fourth part is open source super pack generation and adaptive resolution network layouts, which we usually adapt to the cytoscape that we will going to store in the network database exchange repository called NDEX. And all those network generated and also this validated result from our biologists, we can create the curated data and with those manuscripts prepared to disseminate the result as well as to assess and evaluate the proposed approach. And those unpublished work will just provide the bioarchive link, and for those published work, we're going to share the PubMed ID and also DOI link in front each journal. So in this element 2, the two software and codes, how we going to how we can create the tools in regarding to the dissemination of this code. So for this proposed activities regarding generated data, analysis, result, and publication, we're going to store in the GitHub repository. Especially, we have the AI.mat GitHub repository to be publicly available. And we also have the node that's introduced by Zhandos and bioarchive for those unpublished work. As well, we also introduced the DMMS, which is a model management system. The core part of this DMMS will be a database management system based on the products such like Oracle, and they will store the simulated and real-world SuperPack dataset directly. And for those public available source, we're going to create open access web server with a URL that you have been seen before. And for the data access and analysis, and also we also provide open access web app with URL, such as the Pager web app for enhanced visualization and interpretation of the data. In the element 3 is about the standard. So particular, we have this pack naming convention, include 3 letters plus 6 digits to reflect the data duration, data process, and the type of the relationship to further facilitate pack classification. So for example, here this have AX. The first letter stands for the functional genomics, and the second stands for the curated from literature, and the third X stands for the no interruptions. So therefore, you can immediately recognize what kind of this pack have been classified as. As well, in this super pack, we're going to create this summarized and condensed version of the ontology with a standardized vocabulary to describe those biological process, molecular functions, cellular components, and the phenotypes. In the element 4, data preservation, access, and release timelines, we particularly mention the web server we have provided called Pager 3. They also provide the API for the data access and GOFTP links to download the data. As well, we also provide a download link here for you to direct access to the data we create. And also as well, for those internal data, we're going to store on several servers, such as mentioned UAB Data Center, which has a RAD backup system enabled in areas specifically dedicated to this project. Also, for those unpublished data, we were going to use the password protected limit access to the workstation computers. And here is a picture of the UAB storage area network. And also all the data stored in this SAN will be checked and verified monthly. And the add-on 5 is about the access, distribution, and reuse consideration, which also like all of attendees' interests about the reproducibilities. So we have all these requirements from the NIH and data sharing and archiving policies. We will follow those policies as well, since we didn't use any clinical data, so there's no limitation for sharing those data we generated. And also, all this data will be freely disseminated and under the open source MIT license. We also specifically mention about the libraries we're going to create, especially for these R bioconductors libraries. We're going to store our own libraries here, and for the Python, we're going to use PyPy. In the add-on 6, going to review the oversight of the data management and sharing, which we have these 3 key personnel in this grant. We mentioned about the committees that we're going to form and generate data usage protocol in specific super PAC revision, usage approval, and super PAC sharing to fulfill this external request. As well, we also have this internal review of all those progress generated by our teams, such as those UAB bugs in stage result. We are going to review, monitor, supervise. And as well, we also just have those web interface and also web server. We will going to have a close look at those progress to make sure every element is correct and fully accessible to our users. And this is from my part. So Dr. Chen, you can take over to get this closed.
Yeah. So I think I only have one slide left. So essentially, we want to just ask people acknowledge these resources like Informatic Institute, AI Med Lab, CCTS, and Chia Research Computing. We do have a sheet that's called a Q&A, how to develop this. And it's a work in progress, and I made the disclaimers, and it's our own opinion, but we attempt to address questions way beyond today's lecture. You can access by scanning things here or going back to the URL. So we have just less than 5 minutes left. I've already answered some questions, and I wonder if there are additional questions that we can answer live.
I have a question, Dr. Chen. If we are using I2B2 as data ability of a study for an NIH grant, are we intended to share that data? Can you explain to me what this?
Yeah. So that's a great question. The question has 2 parts, and then one is if you choose I2B2 as a platform, you can have controlled access to any kind of data you stored in there. And that I2B2 is really good for conducting the research throughout the life cycle. And when it comes to sharing it, you may have to develop APIs through FHIR to provide others with a programmatic access to the data. that would usually requires you to budget for that data sharing. If you don't have that resource, one thing is you should look at the NIH program and see if they requires you to upload your data to dbGaP. dbGaP has a capability to have limited access. Or in the case of National COVID Cohort Collaborative Initiative, N3C, they also have provisions to upload de-identified clinical information there for people to do data science. The least case scenario is you can actually create a Zhandos entry to say, oh, I have this data and it send their controlled access stored at UAB's I2B2. You tell people the existence of the data. If there's a need, people will contact you, sign the data use agreement, and access these data through creating a UAB user ID, Blazor ID, and then accessing it.
Very well, thank you.
Any other questions? Yeah, so I think for the most investigators, your research study probably is going to be less sophisticated than what Dr. Zongliang Yue just demonstrated. So I hope that you will get enough ideas and just check out the U-BRITE website and construct your data management and sharing plan for free without additional cost. But if there's a need and your project is complex, feel free to email me and we're happy to help. Thank you.
Jake, thank you very much for this. Hopefully it's been recorded and we can share it with the broader community, because I think, you know, everybody's going to bump into this, and the more we can give them a simple sort of, you know, data sharing plan button, you know, the better. I think, you know, people are kind of freaking out about this, but this has been a requirement for a long time. It's just now being included in a different way in grant proposals. And so we should have been ready for this. But clearly we need to give the real catch is giving people a place where they can put their data and be able to say, my data is going to be here. And, you know, in terms of metadata and documentation, the requirements aren't that clear. So, you know, I think any good effort in that direction is going to be fine. But really the catch is people need to make sure they crossed all their T's and dotted all their I's, and we can give them a good way to do that. So thank you.
Sure. Thank you all.
Yeah, I realize we're at time. So thanks for the great and practical and very real presentation on each issue that will affect all of us. And, you know, feel free to contact when you need more. I don't think this is going to be a one-time thing. There'll be other opportunities to learn more. But if you need any help, contact Jake or the other members of the U-BRITE team. Have a great weekend, everybody.
Good weekend.
Thank you. Bye.
Bye.
Bye.